Bismillahirrahmanirrahim. Today we are going to discuss the autonomic nervous system. We have two lectures, that is 1 and 2, regarding to autonomic nervous system. I will try to cover both in the same lecture. So, autonomic nervous system. First of all, we must know the classification of nervous system. So, nervous system can be classified into central nervous system, which consists of the brain and the spinal cord with specific functions. And then you have the peripheral nervous system, which lies outside the central nervous system, and it can be further subdivided into motor and the sensory nervous system. Then within the motor nervous system, you can have the somatic nervous system and autonomic nervous system. Now, somatic nervous system, as the name indicates, it controls the voluntary muscular movements. And the autonomic, as the name indicates, autonomous on its own. Mostly the functions are carried out as such, and it controls responses which includes fight or flight, which comes under the sympathetic nervous system, and then rest or digest, which comes under the parasympathetic nervous system. And then we have a third division as well, which is called as enteric nervous system, not shown over here. The nervous system of the GI tract. It contains a lot of nerves, hormones, and neurotransmitters involved, which usually works on its own without much integration with the brain or the higher centers. So it is also a nervous system. It has also a nervous system, a complex nervous system, which is called as an enteric nervous system. So reviewing the classification, we have the nervous system being classified into the central nervous system and the peripheral. The peripheral is further divided into motor nervous system and the sensory nervous system. The sensory nervous system consists of sense organs which send information to the CNS, and the motor nervous system usually pertaining to muscles and the glands. So then further this system is further classified into the somatic, which controls only the voluntary muscular movements, and then the autonomic nervous system. Now, the efferent division of the peripheral nervous system, it is usually a communication link by which the CNS controls the muscles or the glands, which are called as the effector organs that carry out the intended effects or actions. So these can be heart, for example, if we are considering it is a cardiac muscle. Heart contains cardiac muscle, and and as for example, we can have stomach for the autonomic nervous system, which is a smooth muscle. So we can have various functions, either delayed emptying of the stomach or intestine, or early emptying. Then you have the respiratory muscle, which is a skeletal muscle, which can help us augmented breathing in response to exercise. And then you have sweat glands, which are exocrine glands, which causes sweating if we are exposed to hot environment or in certain states. And then you have the endocrine pancreas, which causes increased secretion of the insulin, which is a hormone that puts excess nutrients in storage following a meal. So all these examples which I gave you illustrate much of the efferent output, which is and what does it show? It shows that it is directed towards maintaining homeostasis. Now the efferent output to the skeletal muscle, as shown in the somatic nervous system, it is voluntarily controlled, and it is non-homeostatic activity, such as if you are riding a bicycle. Many effector organs are subject to hormonal control or to intrinsic control, and a lot of neurotransmitters, usually they are involved in it. So this is a brief classification of the nervous system, and then the target organs of the autonomic nervous system. I told you it can be a cardiac muscle, it can be a smooth muscle, it can be a skeletal muscle, it can be an exocrine gland, or it can even be an endocrine gland. Now here in this slide, I have shown briefly the organization of sympathetic nervous system. Sympathetic nervous system is also called as thoracolumbar outflow because a lot of the fibers, the preganglionic fibers that take origin, they usually lie within thoracic 1 to lumbar 2 level of the spinal cord. So usually at the T1 to L2 level, the preganglionic neurons, they take origin. Then you have the ganglionic neurons. It can be of different types. For example, it can be paired, it can be unpaired, or it can be in a form of a adrenal medulla. And then the target organs we have already discovered. Now the learning objectives are, at the end of this lecture, you should define autonomic nervous system and classify nervous system. Describe pathways of sympathetic and parasympathetic nervous system, and then enumerate the sites of release of neurotransmitters, and describes the properties of autonomic receptor types. So these are your learning objective for this. As already outlined, sympathetic nervous system is also called as thoracolumbar system because it arises from the lateral horn of the spinal cord from the level of thoracic 1 to lumbar 2. The cell bodies are located at the level of thoracic 1 and lumbar 2. So we have two types of fibers for both sympathetic nervous system and parasympathetic nervous system. We have preganglionic fibers and then we have postganglionic fibers. So the preganglionic fibers of the sympathetic nervous system, they arise from the lateral horn of the spinal cord, and then they leave the spinal cord with the ventral roots of the spinal nerve. Cell bodies usually are located within the gray matter of thoracic 1 to lumbar. Here, I guess we did this. Then, what is the destination of the preganglionic fibers after reaching the sympathetic trunk? Now, these preganglionic fibers can go through a lot of. They have a lot of you can say paths to follow. For example, they either may terminate in the same ganglia that it enters, and or it may pass upward or terminate in another ganglia, or they may pass downward and terminate in another ganglia, or they even may leave the sympathetic trunk without synapsing and terminate in other ganglia which lie outside the sympathetic trunk. So these are the different termination points of these preganglionic fibers after they reach the sympathetic. Now ganglia, what are ganglia? These are structures where synapsing between the pre and the postganglionic fibers occurs. One preganglionic fiber synapses with several postganglionic neurons. Then there are a lot of different sympathetic ganglia. They may either be sympathetic trunk, prevertebral or peripheral ganglia, or subdiaphragmatic ganglia, or paravertebral ganglia. A lot of different ganglias. Now, the characteristics of the postganglionic sympathetic neuron. All postganglionic sympathetic neuron arise from sympathetic ganglia, and these are short, these are non-myelinated as you saw in the diagram, and they are type C fibers, means they would be slow conducting fibers since they are not myelinated. So the postganglionic sympathetic fibers, as we said, may pass through the gray ramus communicantes and re-enter the ventral root to reach the spinal nerves and innervate the sweat glands, the blood vessels, the pyroerector muscle, whatever their target organ may be. So we are going to do one of the slide with the diagrams in which you better understand. Acetylcholine is released at the preganglionic fiber, but norepinephrine or adrenaline is released at the postganglionic. One of the difference between them. And then there's difference of the receptors as well. So all preganglionic release acetylcholine and they are excitatory at both the sympathetic nervous system and the parasympathetic nervous system. And then the sympathetic postganglionic fibers, they usually release norepinephrine and it can be either excitatory or it can be inhibitory depending upon the receptors over there. The excitation or inhibition is receptor dependent. Now this is very important point because when we further do the different types of receptors and where they are present and what changes they bring about, then it will be much more clearer to you guys. and receptor mediated response. For example, synapsing below a level, going up, then exiting as, and if, for example, it thinks that it comes over here and says, oh no, I don't want to go down a level, I don't want to be a part of the white ramus communicants or a gray ramus communicant, and can exit as its own, as a, then it is called as a splanchnic nerve. So it can go down a level, go up a level, or exit on its own, called the different type of ganglia. You can see this is the paravertebral ganglia, the sympathetic trunk, and then the prevertebral, that is in front, on the side, prevertebral in front of the vertebral column, para on the side of the vertebral column, and prevertebral ganglia. Further, there are many different types: the celiac ganglion, superior mesenteric, and the inferior mesenteric. So the paravertebral ganglia are located along the sides of the vertebra, and they are united by the preganglionics into the sympathetic trunks. Now we discussed in the previous slides that much of the effects of the sympathetic nervous system usually comes from, and the parasympathetic, of course, from the types of the receptors it has. For example, for the sympathetic nervous system, if we see, we have alpha receptors and beta receptors. These alpha receptors are further classified into alpha 1 and alpha 2. Now both of them are quite different. Alpha 1 usually follows the GQ stimulatory pathway mechanism, usually brought about by G protein coupled receptors. And then you have the alpha 2, which usually follows the G inhibitory type of the mechanism. So alpha 1, wherever alpha 1 receptor is present, it would cause vasoconstriction because it goes through the GQ mechanism of excitation. Now, GQ mechanism, three mechanisms you must, it's kind of your assignment. You just go through these three mechanisms: the GQ mechanism, the GI, that is the G inhibitory mechanism, and the GS, that is the G stimulatory mechanisms. The GQ mechanism usually works by activating a phospholipase C, and this phospholipase C then further cleaves the phosphatidyl phosphates into inositol triphosphate and diacylglycerol. This IP3, also called as anisotol triphosphate, then retrieves a lot of calcium from different channels, from different areas, and this calcium causes constriction. So wherever there is alpha 1, just make it a mind that it will cause vasoconstriction. So alpha 1 are usually present on the vascular smooth muscle, erector pillar muscle, in the kidneys, where splanchnic circulation in the GI tract, and then two different, these are the bad places for alpha 1. Now, the alpha 2 usually are present on the presynaptic nerve terminals. There is usually a presynaptic nerve terminal and a postsynaptic terminal. Alpha 2 are present on the presynaptic nerve terminal. Now, I told you alpha 1 goes through GQ mechanism by excitating the excitatory effect of releasing calcium through activation of phospholipase C and causes vasoconstriction, whereas alpha 2 follows GI inhibitory mechanism, which causes inhibition of the CAMP mechanism, decreased ATP formation, and would cause inhibition. Now coming to the beta receptors, you have beta 1 that are present on the heart, in the heart, and the juxtaglomerular complex in the kidneys. These are the two places where beta 1 receptor is usually present. Now beta 2, this is the beta, we're talking about the beta 1. Beta 2 usually in your respiratory, in your lungs, it is usually present. And then you have the beta 3, which is usually present on your adipose tissue. So wherever there is beta 1, beta 1 usually it follows the G stimulatory pathway. Now the G stimulatory pathway usually excites the system, or the formation of ATP through activation of CAMP and the formation of protein kinase A, and therefore cause excitation. It is the stimulation. So it follows the G stimulatory pathway. And the beta 2 usually, it is usually present on the vascular smooth muscles, which we need usually to be relaxed. As I said, it is usually present in the in your lungs, and beta 1 is usually present in your heart and kidneys, and beta 2 usually on your vascular smooth muscles, which usually you want to relax, and beta 3 is usually present in the adipose tissues. So this is a relative location, though for different organs. For example, we have different regulation of these receptors which are present in different muscles. They can be both, either they can be a single alpha, or they can even be innervated. For example, in heart you have alpha 1 as well as beta. So where you want to constrict them, so the alpha 1 usually occurs, and where you want to relax them, so usually have the beta 2 to relax them. So now this is the relative explanation of what we already done. Alpha 1 are located on the vascular smooth muscles of the skin, the splanchnic region, the GI, the bladder, and it usually produce excitation. Now it produces excitation because it follows GQ mechanism of activation. And then we have alpha 2, which are located on the presynaptic terminals, and it often produces inhibition. It produces inhibition because it follows the GI system of uh, activation of the. Now, the beta 1 relative location in the heart is on the sinoatrial node, the AV node, atrioventricular node, and the ventricular muscle of the heart itself. And we said that the B beta 1 usually follows the GS mechanism, the G stimulatory mechanism. So, of course, it would cause excitation. Now, excitation in heart means positive inotropic effect, means increased contractility. The heart would contract more forcefully as compared to the previous situations if it is activated. Then positive chronotropic effect means increased heart rate. For example, if somebody is having a heart rate of 72, if we excite them, or the sympathetic nervous system is activated through beta 1, so the heart rate would go from 71 or 72 maybe to 90 to 100 or something above that. Positive dromotropic effect of the heart means increased automaticity of the heart, increased generation of impulses from the impulse generator, that is the sinoatrial node. Now, these beta 1 receptors are sensitive to both the norepinephrine and epinephrine, to both of these. Now, coming to the beta 2, as we said, lungs, we did one, that is, for example, in the lungs, it is usually present in the bronchial smooth muscle, and then and we discussed that it is present in the vascular smooth muscles of different tissues, for example, the skeletal muscle, the GI tract, and the bladder. Now, what does the beta 2 do? It causes relaxation. So for example, dilatation of vascular smooth muscle, dilatation of bronchioles, and relaxation of the bladder wall. They are more sensitive to epinephrine than to norepinephrine. So these are the adrenergic receptors, beta 2. What they are usually going, when they are excited, they would cause vasodilation because of relaxation. And whenever we say excitation, we mean vasoconstriction. Constriction, because a lot of calcium is released, the blood vessels would constrict. In decrease in size, constrict. And relaxation means dilate, increase in size. Now, this is a very good review of what we have done till now: signaling pathways and mechanism. What are these mechanisms? Guys, you must go through the GQ mechanism. I told you, through the activation of phospholipase C, then the conversion of phosphatidyl diphosphate into phosphatidyl triphosphate, inositol triphosphate, and diacetyl glycerol, and IP3 then further releases calcium. And then the GI, which would inhibit the CAMP mechanism, the GS, which would stimulate the CAMP mechanism, and the G inhibitory, of course, decreasing. So these are the three mechanisms which you should go through. Now, one thing over here is, you can see the receptor type. You can see heading adrenergic and cholinergic. Now, adrenergic means if the neurotransmitter to which these receptors are susceptible or more excited to are norepinephrine or epinephrine, for example, noradrenaline. So these are called as adrenergic receptors. And if they are susceptible to acetylcholine, these are called as the cholinergic receptors. And these are further classified into NM, NN, and M1, N2, and M3. Now, these cholinergic receptors we usually, for example, have classified into nicotinic receptors. Nicotine, we have given the name because it is a compound found in tobacco. Nicotine, it produces the same effect as this would. So that's why we have given the name nicotine. Or muscarinic receptors, which is related by M. This is because the type of a mushroom poison or poison found in some frogs, I don't know which species, which is called as muscarine. These receptors are susceptible to them, so we have given them the name of muscarine. They would bring about the same changes as would acetylcholine. So these are the sub names that we have given them. So NM, that is, they are found on the muscles, as M say, that is skeletal muscle. And these N receptors, that are nicotinic receptors, these are ion gated channels. Now you can see here written mechanism is opening of the sodium potassium channels, opening of the sodium potassium channels. So these are the nicotinic receptors are ionic type of channel receptor, ligand gated receptors, whereas the muscarine are the G protein coupled receptors, which we have already discussed, that is GQ receptor, GI receptor, or G stimulatory type of receptors. So the relative presence you must know, and the classification as well. NM on the skeletal muscle and NN on the autonomic ganglia. They are usually found. Then you have M1, M2, M3, M4, and M5 as well. Since they are not very important, the 4 and the 5, they are not mostly outlined here, but they are usually found in the CNS. Now M1 in the CNS, usually what would be their function in the CNS, that is, it would help in memory, consolidation of memory, and then certain other functions. Then M2 usually present in the heart. You can see the mechanism is GI, that is decreased cam. Now you can see these are the G protein coupled receptors, all of the M receptors. And then you have the M3, which are the glands present in the glands and the smooth muscles, which follows the GQ pathway. Now, if you remember, if you go through a sequence, for example, if you remember that M1 is always excitatory, M2 is inhibitory, so M3 is excitatory, M4 is inhibitory, and M5 is excitatory, so it would be easy for you to memorize them. So these are the relative presence of these receptors, and usually where they are found in the glands. I told you M3 receptors, these can be exocrine glands, lacrimal glands, salivary glands, sweat glands, pancreatic acinine glands, gastric glands, and God knows a lot. And M2 usually in the heart. In the heart, it goes through a lot of, like, for example, the SA node, the AV node, the bundle of His, and M1 in the CNS, a lot of places. Now, when we were doing the dual innervation from both the sympathetic and the parasympathetic nervous system, there are certain exceptions in this general rule of dual reciprocal innervation by the two branches of the autonomic nervous system. Number one is the blood vessels. Most arterioles and veins, they are innervated. They receive only the sympathetic nerve fibers. And regulation is accomplished by increasing or decreasing the firing rate above or below the level in these sympathetic nerve fibers. So the only blood vessels to receive both sympathetic and parasympathetic nerve fibers are those supplying the penis and the clitoris. That is only an exception. Otherwise, the blood vessels usually have only sympathetic innervation. Now, most sweat glands, they are innervated only by sympathetic nerves, and the postganglionic fibers of these nerves are unusual because, as we said, that the postganglionic sympathetic fibers release norepinephrine, but this is an exception to it because it releases acetylcholine. In such case, these are excitatory. Now, the salivary glands are innervated by both that is the autonomic and the parasympathetic and the parasympathetic, but they are not antagonist in this case. For example, if we said heart sympathetic increases heart rate, parasympathetic decreases heart rate, this means antagonist, means opposite to one another. In most cases where there is dual innervation, sympathetic and parasympathetic, they are opposite to one another. But in the case of salivary glands, they are innervated by both sympathetic and parasympathetic, but they are not antagonists. Both stimulate the secretions of the saliva, but the difference is in the saliva's volume composition. It differs depending upon which autonomic branch is, for example, dominant. Either it is a sympathetic nervous system and a parasympathetic nervous system. And then we have, you all know what is adrenal glands, right? Adrenal glands, they have a cortex and a medulla, and the adrenal medulla is a modified type of a sympathetic nervous system that does not give rise to postganglionic fibers, but it releases epinephrine or adrenaline and works as a sympathetic organ. So this was, I guess, and if you have any queries, you can email me uh, through, or you can even WhatsApp me. And best of luck. So meet you next time in another lecture.